Hello, everyone, and welcome to our new webinar. What I'm going to do a little bit, wait for a few seconds for everyone to join before we start this webinar to give people some time. And we are very excited to give you the latest on the updates for the vegetable systems trial project that was started 7 years ago, and other information gathered during this time on new area of research. So it is going to be a dense webinar for you. Okay. Let me see here. And I think we are going to start right away. Just few housekeeping points for you. You can hear me, and if not audible, please use your audio settings on your screen. And if you have any questions, I appreciate that you include them under the QA and not in the chat, because that a little bit disturbs the presentation. In the QA, list all your questions and we will try to answer them at the end of the presentation. And if you have any problems of connecting or not seeing or hearing, please call Justin Barkley at the number 610-683-1442. And if you have an issue of not able to call, use his email, justin.barkley@rodale institute as one word .org. And this recording for this presentation would be available on our website in a week from now. So the outline for this presentation is about giving you a little bit background on practices that led to degradation of soil health and nutrient quality. And that's majorly I do for new attendees who joined lately this webinar and does not know much about vegetable systems, right? I will share with you some of the results that we gathered over 6 years on soil health, as well as nutrient density on 2 major crops across 3-year crop rotation. Also, the second part of my presentation will entail talking about ergothioneine. And some of you have heard of this, some of you not. Ergothioneine is a rare amino acid, and it would be an interesting topic to share with you and some of the trials that we had at Rodale Institute. Definitely, I will make more details information about the second part so people can understand what is this amino acid. At the end of the presentation, we'll have a Q&A session. And for that, I would be closing my video to make more of the focus of this work. So let's see here. All right, for those who have joined us recently and do not know much, or we want to refresh your information, the vegetable systems trial has been initiated since 2016 as a long-term research study. This is established at Kutztown, Pennsylvania. It is the headquarters of Rodale Institute, with the aim to compare side by side the vegetables that grown in organic and conventional systems. And within each of these cropping systems, we included intensive tillage and reduced tillage. So we will understand how management practices along with the cropping system will impact the soil health and the nutrient quality of the crops that we select to grow. When we talk about intensive tillage, we talk about using intensive machinery and repetitive and frequent cultivation throughout the growing season. When we look at and talk about reduced tillage, it would be in the organic system to use a less intensive equipment, but in a way that does not disturb the soil, but usually at the beginning of the season in spring after cover crops are up and ready for rolling crimping, we use a equipment called roller crimper. And the ones that we use in the conventional, instead of rolling crimping, we just burn down the cover crop with a glyphosate, is a herbicide. And these equipment and practices are to mimic the big scale growers. Just to mention also in the intensive tillage, when we turn down and incorporate the cover crop with equipment like moldboard plow, we prepare beds and we put plastic like you see here on this side where we apply plastic there as a mulch. The crops that we select, these are beans, lettuce, potato, squash, winter squash, and sweet corn. Each of those represent a habit of either upright or vine or root or leaf or a fruit. And these were selected because they fit in a crop rotation of 3 years. And also they fit in the system where when you prepare them or seed them or plant them, they can come out nearly at the same time in the fall. And also considered economical crops for Pennsylvania and the Mid-Atlantic. Now, the question why we are doing this, because literature have shown there are, with time after World War II, there were measurements on fruits and vegetables which showed decline in these nutritional components, minerals, proteins, but there was increase in starch. And this is because there were, after World War II, the breeding programs expanded and start to look into increasing yields, increasing size of the fruits or vegetables, and increasing more of the sugar or starch in these crops. But that took an impact by declining more of the other nutrients. So this brings the question, is our food or our crops that we grow since then are really nutrient dense, or they are just full of sugar and starch and calories? The other factors that also happened during the era after the World War II is the implementations of heavy equipment and frequent plowing of the soil that led to more of decline and degradation of the soil by carbon loss to the atmosphere when they are exposed to soil microbes, and we start to have more emissions of gases such as carbon dioxide and other gas emissions like methane or others. Also, the more you plow frequently, you are degrading the soil from the quality of having aggregate stability. And also, the less aggregates there, the more your soil would be prone for erosion when there is a downpour of rain. And when we have more of practices that include a lot of spraying, either herbicides or pesticides, or adding more of the soluble fertilizers, we are adding more to the pollution, either the air or the water, and of course, hazards to the human health. So based on all these factors, VST was established, and we wanted to compare side by side how we are linking the soil health to the food health and to human health. This is a long-term project, takes time to link those components, but definitely this is the major goal of this trial. So let us look at some of the results that we have gained throughout the time. And here I'm gonna talk about the first layer of soil, that it is between 0 to 20 centimeter, and this is soil carbon. And mainly we are looking into how soil carbon changed with time, from time of establishment and compare it to the time in 2022 after 2 crop rotations have been done. Soil carbon is very important because it is important in building the quality of the soil, and it's one of the soil health indicators. So let's look at the soil carbon here. And in this graph, you will see that the conventional and the organic. And in the conventional, we have both black plastic and reduced tillage. They are in red color, and black plastic intensive tillage with reduced tillage in blue. So in 2016, we have those all similar. And then when we compare soil carbon to 2022, we could see a major decline in soil carbon in the conventional. You could see that decline happened in the reduced till and more in the black plastic intensive tillage. However, the good news is that when we looked at the organic system, 6 years after establishment, we found that we have increased by 1% the carbon in the reduced tillage organic, and we are still at the same level of carbon, so we are not building there in the intensive tillage black plastic organic. Looking at organic matter, similarly, we are taking the same color of bars and what it means. So here, if you look at 2016, in these 4 bars over there, they were all the same. Of course, this is homogenized soil at the beginning. And then when we move more to 2022, you could see again that we have declined more of the organic matter in the intensive tillage conventional. However, we built over in the reduced tillage of the organic system. We did not build more, although it is there, but it is not statistically significant from the time we have started in 2016. So it is, yes, it is significant, but not as significant as those in the reduced tillage, what I meant to say. The increase, it was statistically significant by 1% of organic matter, and that was in the organic reduced till. It took 6 years to see this increase. And in 6 years, we found the decline as well in the conventional, as you could see here. But that increase is favorable for us because that gives you more of the stability in the soil. As climate changes and less water would be available, or less rainfall would be in certain areas of the world, you could see that every increase in soil organic matter will help the soil to hold more of water, about 20,000 gallons of water per acre. Meaning when plants are exposed to difficulty of less rain, that soil is your buffer capacity, that it is really holding that water for the plant to continue slowly and taking that water. That's the great advantage of having increase of the soil organic matter. If we are looking at the protein in the soil is important because it is the building blocks of soil health, and it's another major indicator. It is important because it depends on the nitrogen pool in the soil, and the nitrogen is sensitive to management practices. So we look here again, and we see in the 0 to 20 centimeters, we see that at the beginning they were all homogeneous, same level, but we have seen a decline in the conventional, especially in the intensive tillage, but a big rise in the protein in the organic reduced tillage than in the conventional. That increase in the protein was about 50% from the beginning in 2016 and was statistically different. This helps the protein to sequester carbon as glycoproteins, which has a lot of more time to degrade rather than quickly going away. So the message that I leave with you here on this section is that yes, we can build soil and build health, and remember the word building, and it's not just putting something and taking it at the end of the season. Here we are building, meaning every year we are adding and keeping that quality of soil improving. So we can enhance it by implementing organic systems and reduce tillage. Not only organic by intensive tillage, because organic intensive tillage is like taking your check and spending it at the end of the at the end of the season. You can also decline the soil by implementing different systems, which is the conventional, irrespective whether you're doing it reduced tillage or intensive tillage. Okay, so let's say we have improved the soil. Does this mean we are enhancing the nutrient quality? Are we enhancing the nutrient density in the crops? That's a question. It's a very big question. It takes a lot of work to answer it, and let's take step by step to understand it. So let's simply define what is a nutrient density. It is a relationship between calories and nutritional quality of the food. Meaning, if I have more of a nutrients relative to calories in that food, this means it is nutrient dense. On the other hand, if I have the other way, where I have more calories than nutrients, then I am having a deficient crop or a nutrient deficient crop or food. So it is all about, if you remember, we talked about the breeding systems that really made more of the use of breeding high yielding and more stuff. Excuse me for that. We are here going to look about nutrient quality in a way that really tells us whether the crops that we are producing in the vegetable systems trial is improved over time due to the improvement in the soil health, or we are having it less and less going down. But to have it according to one standard, just taking the nutrient itself concentration from the lab and just report it, oh, we have more in the organic than the conventional, we really need to relate that to the energy, the calories that the crop usually produces. And also, we have to relate it to the daily nutrient requirement by the adult. And so the USDA have published tables on what is your daily intake should be for an adult or for a pregnant woman or a child or different genders, like female or a male, different ages. And so we use these data or these tables to prepare what we call the nutrient quality index. But also we assess the energy that comes from these, which is the calories from these crops. And so in that case, we prepare the equation and we assess. So usually if it is standardized to be equal, one or more than one or less than one. So if the index is greater than one, meaning the diet, if that was entirely from that crop itself for that day, the daily nutrient meet or exceed the recommended daily intake rate for that nutrient. So supposedly I'm looking at calcium, or I am looking at protein, or I'm looking at iron. So if my food was only potato on that day, all day, and my nutrient requirement, let's say for potassium, is 4,600 milligrams, then I would know whether putting that number into that equation and see if the potato is providing me with potassium enough or not based on where it was grown. So if it was grown in the organic, is it different when it is grown conventionally, or if it is grown in the intensive tillage versus the reduced tillage? And remember, those may vary from year to year. So if it is less than one, then meaning that crop or that food that I am taking is, we call it less, did not meet the daily intake for the nutrient that we are assessing for. So I'm gonna go into the results now, and here is the squash, winter squash. So this is overall mean nutrient quality index according to the cropping systems. And this is over 3 years data, so overall mean. So the elements that we assessed, it was phosphorus, potassium, calcium, magnesium, iron, manganese, copper, zinc, vitamin B6, vitamin C, and proteins. The systems, conventional and organic. In the numbers, NQI, if you remember, if it is 1 or equal to 1 or more than 1, meaning these are considered nutrient sufficient. So in this case, if I look at phosphorus, yes, it is a nutrient sufficient because it's more than 1, either in the organic or conventional. So until now, between 2020 and 2022, phosphorus is still the same in that crop. Similarly with potassium. Calcium, we start to see significant differences here. And so we saw more of the calcium start to increase in the organic, and it was less in the conventional. But what it would be really nice to see in the coming crop rotation between 23 and 25 and adding all those, have we seen a less in the calcium and more in the organic? And similarly, I could say for iron. So iron here is less than 1. What the NQI is less than 1, meaning that this crop itself, the winter squash, is not as what you call it, nutrient dense in iron, but it is a nutrient dense in other elements. So I would not depend on winter squash if I have some iron deficiency in my blood. So I need to go into another crop that has iron there. And that's where the NQI really helps to understand if there would be measurements or practices need to be done how we can alleviate and change those nutrients in a way that we can build these nutrients over time, that the crop itself at the end, it will be sufficient to give you that nutrient instead of going to crops that they are fortified with elements or nutrients not necessarily done naturally by the plant. So here you could see that the copper in the organic was higher, the vitamin B6 was higher, and proteins was higher in the winter squash. When we looked at intensive tillage, which is represented by BP with black plastic and RT, reduced tillage, we start to see more variation between the two practices, more of the phosphorus in the soil, in the crop, meaning the phosphorus is more coming and taken up by the plant in the reduced tillage. And that could be due to soil microbes that are available and they have like fungi that they are expanding their mycelia and then they are bringing more of those nutrients to the crop. And so that could be more in the reduced tillage than in places where we have intensive cultivation throughout the season. Similarly, potassium is increased and calcium here is increased in the reduced tillage, copper, and unlike the others, we have vitamin B6 showing, in this case, it is more in the intensive tillage. And you may ask why, and that's because in the structure of the vitamin B6, we have nitrogen. And nitrogen is always mobile, meaning that the more you incorporate things, the more you cultivate, those microorganisms will work on the nitrogen and make it mobile and more available. And so if the structure of the vitamin B6 has that nitrogen, that is expected to see more of that vitamin B6 in the intensive tillage. However, as I said, if we continue to do this, and combine the data in the coming few years that we get, it would be really interesting to see the fluctuations and the stability or the increase or the decline of those different nutrients and understand how management of the soil will really impact the nutrient quality. Let's move to another crop now. It is the potato. Potato, similarly, we did the same with the same period of time. We used Lehigh potato. It's very common in Lehigh Valley here in Pennsylvania. And so before I start, I just want to bring to your attention the protein availability in the potato more than in the winter squash. If I just go one more here, you could see the winter squash coming almost to be 1, but here in the potato is more than 1. So those elements that are more than 1, the NQI, more than 1, meaning they are satisfying the needs for that daily intake with that measurement of the calorie. So we are good in phosphorus, no differences in phosphorus in potato between organic and conventional, but we start to see significant differences in the organic for potassium, magnesium, copper, vitamin B6, and proteins. Here we start to see variations. I expect in the coming few years, these numbers will start to widen, the difference between them, they will start to widen. And if I look again here to calcium, potato is not a source of calcium, unlike the winter squash, where if we look back here, you could see that in this system, that winter squash was meeting the needs of the daily intake of calcium. So iron is becoming closer to 1 in the potato. It would be interesting to see this in the coming years, what would be that index. Looking at the reduced tillage and black plastic, we start to see that the potato itself here, that it is more in the intensive tillage black plastic, in the phosphorus, unlike the winter squash. As I said, the different crops, they have a different way of nutrient uptake, and the way they are, the root system will be working with the practice in the soil and the microbes may give us different analysis, meaning they will give us different results. This doesn't mean that in, let's say, 2020 or 2021, the reduced tillage was higher than the black plastic, but this is average over 3 years and multiple replications. So it is more rigorous work that these results came out from. Here we start to see iron is getting better under reduced tillage, and vitamin C is really becoming higher and greater in the reduced tillage. So information is very valuable because we want to see if we will be recommending in the future for growers and lands to be cultivated and be producing potatoes or winter squash or other crops. It would be helping to know the method that if we are targeting a certain nutrient, we need to increase, and which crop will really be benefiting from the management practice we implement to increase those nutrients. So a summary of this part one is that after 6 years, our results show that the combined effects of using organic cropping system along with reduced tillage management practices will enhance soil carbon, organic matter, and proteins in the vegetable systems trial. And this will have a effect on the soil microbes and also on the quality of the soil to be resilient for any challenges, environmental challenges in the future. When we looked at the index of winter squash, we found that the winter squash increased in the organic system with the minerals, vitamin B6, and proteins. In the case of potato, we only saw potassium and magnesium and vitamin B6 and proteins with increases in the system with the organic, but the iron and vitamin C was impacted by the practices. So now I'm gonna talk about a new area of work that we have been looking at different crops in VST and outside VST, and it is related to a rare amino acid. It's called ergothioneine. And in short, we are going to call it ergo. So let's define or talk about some of the facts of this amino acid. And that is the amino acid ergothioneine is called longevity vitamin. And it is discovered back in 1909 in a ergot fungus that was grown on rye, a cereal rye. And then the scientists who discovered it found that it is very stable antioxidant, and it is produced by certain fungi, cyanobacteria, and some bacteria. Now, there were no much work done on which fungi. There are trillions of fungi and bacteria, but now we are teaming with scientists across the United States. We are working on pinpointing which of those fungi or bacteria Apologize for coughing. Okay, so what does this ergothioneine do? It is an antioxidant. It is used as a regulator for anti-inflammatory. It is helpful in protecting our body from oxidative stress. It neutralizes all these reactive oxygen species, such as hydroxyl radicals and hydrogen peroxides that are produced under stress. So meaning it has a big effect. Just to mention that ergothioneine is produced in soils when it was found, in soils and in crops such as the rye, in crops and soils that are mostly that have less disturbance, meaning that mushrooms grow in these soils. But also they found them in mushrooms, meaning mushrooms contain ergothioneine as well. So people who are depending or consuming mushrooms, they would expect that they have ergothioneine, this rare amino acid, in their diet when they consume mushrooms. And for us as human beings, we cannot just make this amino acid in our body, but we have to depend on either consuming an animal or a plant that already have taken into them that ergothioneine. So the plant doesn't make it, the animal doesn't make it, but our diet that we take is either a plant base or a animal base would be grown in an area that ergothioneine is already in that soil. So I will share with you some of the results that work that we did in potatoes as well as asparagus. Potato is an annual crop, asparagus is a perennial crop. And so in the case of potato, we were trying to see if mycorrhizal fungi is one of those fungi that inhabit the soils and whether they can participate or contribute into the ergothioneine. So this graph shows on the y-axis ergothioneine concentration in potato, and on the x-axis the mock, meaning that the medium that we grew the potato in is sterilized 100% and no addition of any mycorrhiza in it. The other treatments here, this means the media has been sterilized, it was a plant-based, and then we added inoculums from mixed species of fungi, of mycorrhizal fungi that came from the organic soil in the vegetable systems trial, and then other single species that were established at USDA and inoculated the soil where the potato was grown. Definitely you could see that where we did not apply any of the mycorrhizae, the ergothioneine was less than 0.1 milligram per kilogram, whereas when we applied the inoculum that has the mixed species of fungi from taken from vegetable systems trial, organic system, it really gave at least 0.55. It is like 5 times greater than without it. And then when we use the single ones, it really did not give the effect as the mixed species. This work has been published along with other colleagues, and it is published in the American Journal of Potato Research. The other experiment here is about asparagus. you would see also ergothioneine in the y-axis, and on the x-axis, again, uninoculated, which is meaning the mock, and then the mixed species like we did in the potato, and the other species of mycorrhiza. Again, we use 3 different types of medium here, and the white bar represent peat moss. All these are sterilized when they started. And then the gray bar represent plant-based compost, like we started on this one here in the potato. And the compost is the black bar. Mushroom compost, we wanted to see if we add the inoculum with the mushroom compost and compare it to peat moss or plant compost, how the ergothioneine will change. So if you look here at the mock, in any of those, we have very little, which is less than 1.1, like we have here. And then when we applied the inoculum from the vegetable systems trial, and the mixed species of mycorrhizae, and it continued to increase with the plant-based, and then it also continued to increase when we added the compost. So if I compare potatoes and asparagus, this is what the plant base is giving us about 0.8. And the potato is giving us about 0.55. So that's where in the plant base was given in the potato, and in the case of asparagus, it was a little bit greater. But if we push more with mushroom compost, that ergothioneine was much higher and pretty significant when we compare it to the other ones. However, what it tells us also, if we just not apply any of the inoculums, it will stay, the ergothioneine be very low, meaning the addition of mycorrhiza play a great role in enhancing this ergothioneine and uptake, and works together with the plant roots and uptake of these nutrients. Another project that came from vegetable systems trial, we looked at the crude protein and ergothioneine. In this case, I was looking into the effect on black beans versus green beans. So organic is blue and conventional is red bar. Now here you could see the crude protein is about 23 in the conventional, but it is greater than 28 in the organic. There was a significant difference between organic, meaning organic is much higher crude protein in the black beans. Similarly, green beans were higher, but the crude protein in green beans varies between green bean and black bean. So when I want to look into which of those beans that we have, green bean or or black bean, definitely black bean, consuming more of the black bean will give you more of the crude protein than if you are consuming only green beans. So you could see that organic really favored increases of crude protein. But how about ergothioneine? Let's look at ergothioneine. Again, ergothioneine was in the black bean about 2.5, which is a good number for beans to have ergothioneine. Remember, potato was like 0.5. This is 2.5. There was no effect of being organic or conventional. And remember, this is over 2 years project. When we looked at green bean really benefited from having more ergothioneine than in the organic than in the conventional. That means implementing more of organic green beans, we could go and increase that level of ergothioneine. That's another indicator that how a cropping system improves crude protein as well as ergothioneine. Currently, I'm working on putting this research work into a publication, and hopefully it would be done by the end of this year, and next year it would be online. Another project I worked on, and I thought it would be interesting for researchers and growers, because we talk about ergothioneine, different varieties of beans, different varieties of potatoes, and people were saying, okay, which one I should be using? Or is it different if I use different biological additive? Is it really fungi alone will make the thing, or should I use different biological additive? So I prepared a pot experiment, and I did use, again, the inoculum from the vegetable systems trial, and I compared it to the mushroom extract, rhizobium. This is the rhizobium which we add to the beans usually, or without the rhizobium. It's a bacteria. And then it helps in symbiosis of nitrogen, spirulina, and they kept the control as vegetable systems trial soil alone. Now, people who do not know what is spirulina, it is made of blue-green algae. It is ground and put and packed, and this is a powder. I use the organic powder of spirulina, and it is pretty well known, packed with proteins and antioxidants. I wanted to know whether any of those treatments will really make a difference on the ergothioneine. So let's look at black beans in this case. Again, these are the treatments, and you could see the winners were mycorrhizal inoculum and the spirulina in this case of the black bean. And so this is a pot experiment. If you remember when we talked before on the field experiment, the beans was about 2.5. This is about 1.2 to 1.5. So usually field experiments give you more of nutrients than when you are doing a short-term project in a pot. Now, comparing this to the green beans, we found that the winner was the inoculum with the mycorrhizal fungi, followed by spirulina. And you could see that any of those were less than 1 nanogram per milligram or milligram per kilogram, but black beans has more of the ergothioneine than what green beans has. But mycorrhizal inoculation still was the treatment, biological treatment that could improve when you compare it to only vegetable VST soil alone in both green bean and black bean. This is really a eye-opening, because instead of doing large scale experiments, field work, and spend years, these pot experiments really can pinpoint more for us how we can tailor and focus our research to get because became those as a foundation that we build on it to look into more focused objectives when we run the experiments. And growers would say, okay, now you are finding ergothioneine. Does potato have it? When you looked at Lehigh potato, you found that if you add mycorrhiza, you can improve it. But what if I am not adding any mycorrhiza, but I have different species of potato I'm producing? So we looked at the different varieties of potato, and these are all organic. And so you could see here in the red gold was the highest, as well as russet potato in ergothioneine, followed by fingerling and blue Adirondack and Yukon gold. These two, red gold and russet, had the highest ergothioneine without adding any additive or mycorrhiza or anything. So this is our first step of understanding where these varieties, they fit, or where we may have more or less of ergothioneine. But I want to point out to you that despite we have more ergothioneine in these two varieties, we have other nutrients, such as Yukon Gold, have higher carotenoids than any of the red gold or russet. And similarly, when I look at blue Adirondack, which has the blue skin and blue flesh, or nearly purple, you could say that it has another nutrient, anthocyanin, that it is the highest in this one, more than red gold or russet or Yukon gold, which in Yukon gold is not there at all. So each variety, I always tell to the growers, each variety has its treasure. The treasure either of having more of the protein, or having more of the anthocyanin, or having more of the carotenoid. And in this case, we have different ones having ergothioneine. This means you can still grow all these and you can target what you want to sell it for, but also as a consumer, it's very important to tell you that now we are finding this amino acid in different crops and different varieties. But when you eat, it's better to diverse your food and eat from all these varieties, because you can reap more benefits when you mix all these colors. Excuse me. I apologize for coughing again. So to summarize this part, we found that inoculating mycorrhizal fungi, potato and asparagus enhanced ergothioneine. The different varieties of potato may show different levels of ergothioneine, and the work in any of these pot trials, it is one way of assessing and give us insight for developing future field work. And mainly is to keep in mind that the more we have ergothioneine in crops, the more of the crop resilience and defense mechanism is, and also consuming a variety of food that are high in ergothioneine will increase our human immunity system. So the bottom line is human health stems from soil health. This is very important because this phrase, it tells you that how critical the soil management is for human health. Investing in regenerative organic system and in sustainable practices, it really should help restore soil health. It should promote healthier systems, diversity in soil microbes and food, as well as improve human health by providing nutritious food, reducing exposure to toxins, enhancing natural disease resistance. And today, it is the World Food Day. With all that information, I feel like we are setting up this webinar on a very important day, which is the World Food Day. This is every year the FAO celebrates this day, saying that food is life, hope, and health. And food is diversity in nutrition, affordability, and safety. And so when we implement practices or investigate research, we always keep in mind how we can bring more diversity into the systems, how we can make food more nutritious and affordable and become safe because it has less toxins and it has or not at all pesticides in it, how we can have the ecosystem more resilient. And so I leave you with this message that we all should work towards having more of our ecosystem, our food more nutritious and affordable and safe. I want to thank also the research team who worked with me on several projects, the operation team in the field, and the interns. Not to forget also the funding agencies that provide insights and funding for all the projects, including Grantham Foundation, Pennsylvania Department of Agriculture, USDA projects funding, and Orange County Community Foundation. And with that, I leave you with a important message that take care of your soil, because your soil is the habitat for your food and your food is important for your health. And at the end, soil is important for health and human health stems from soil health. I'm going now to open my video. And let me see if I can do that. Now I'm going to stop sharing. See, yeah, and see if we have any questions. Maybe I should not. Maybe I should not. Let me see how I do that. Hold on. Okay. Yeah. I'm going to look at the. I can't find that. You're sharing. Okay. Let's go over these questions. I did get any PPT or PDF notes of summary of the session for further studies. As I mentioned earlier, this webinar would be uploaded in a week, but we cannot give a presentation PPT or a PDF. Let's see another one here. What is the method used for quantification of soil protein? It is a method used by Cornell Soil Lab. If you Google Cornell Soil Lab, you will find the approach and the method they use for the analysis of soil protein. Good question, Evans. I would like to know the same. We answered that. Nicholas Amanding. Hello, Nicholas. Yes, thank you for your note on nutrient density. And hello for everyone in Uganda. Thank you for joining us today. It is a pleasure. And who else here? Also, my sister is on it, and she said, thank you for an amazing information. You are very welcome. Steven said the blue Adirondack potato having lower ergo values is interesting. I thought antioxidant-rich varieties would have higher levels of antioxidant. I agree with you, Stephen. I was expecting because it has more anthocyanin, but it has more anthocyanin antioxidants. So antioxidants could be different in different classification and in different varieties. So when I have anthocyanin antioxidant, highest in the blue variety, but none in the Yukon Gold. So the carotenoid also, or any other antioxidant, like in the red gold, you will see it has a little bit of carotenoid, but has more of antioxidant ergo. So that's where, when we look into things, sometimes we assume something, but it gives us more insight on what and how and more to dig into. Very good observation. Ivan, do you think AMF and ergo results are soil dependent, as in the results may vary in different soil type? When you say a soil type, I would say that possibly soil type, but mostly it is the habitat of the soil microbes in that soil. So if you look at undisturbed soil like forests, or in soils that has less tillage, or soils that have been building the organic matter in it, where most of those different bacteria and fungi are growing, you will think is more of that soil quality or soil types, depend of how you call it, is having more of potential for increasing the ergo or the AM. But soils that do not have enough aggregate stability, soils that are deficient in clay or deficient in what you call it, organic matter, I would say they are not having the network and the connection between the soil particles to allow to go through. But it could be certain species of mycorrhizae that are different from what we have here, could grow and proliferate in the soil, in other soils. And the short answer, yes, they depend on the soil type and soil quality. Okay, and let's see if we have, there are maybe other ones here. How we are doing on time. Okay. Questions, answer. Okay, there is, do you think it's all dependent? Yes, we answered Evans and and we answered Stephen. Thank you both for putting them into areas. And with that, if you have any other question, you have my email below. And you could email me if any question raised after this presentation, and it would be uploaded in a week. And thank you for your attendance. And I hope I enlightened you in a way to think more about your soil, about your land, about your health, and how you can grow and consume better food. Thank you very much, and celebrate the World Food Day. Thank you. Bye-bye.